For most of you working with other families, perhaps, you'll have some of the same characteristics that I'm exhibiting here. But this item number 6, I think probably no other family, including Orchidaceae, would have the same percentage of novelties that we have in Araceae. Do what? Oh. Could you do it? I mean, I don't see what you mean. Oh, because otherwise it's just going to have one slide? Oh. All right. I've already mentioned that. The Neotropical Araceae range basically from northern Mexico to Uruguay and northern Argentina. They're diverse in every part of its range, but the diversity is most important, high species diversity in lower parts of Central America and northwestern South America. In Central America, the diversity decreases from Mexico as you approach Middle America until you get to Costa Rica and Panama. Then it increases dramatically. The number 827 species are for all of Central America, and note that number is less than the number for just Colombia alone. So you get some idea of how diverse that corner of South America is. Keep in mind that the Guianas, which is much larger than Ecuador, has only a third of the species of Ecuador. Aroids are on this foliage, on the Kuchler map, are in this dark green chocolate color, covering much of the Amazon basin and western slopes of the Andes. But on the Holdridge life zone map, you'll see a lot more diversity in terms of life zones. These maps are very important for predicting where things will be, and the species are richest in the wettest areas like tropical wet and pluvial forest. You can see that Colombia is diverse for the following reasons. It has all these 3 mountain chains with interrupted rivers. It has drainage into the Orinoco River Basin and the Amazon River Basin. It faces the Pacific and the Caribbean. It's just ever much larger in diversity than Central America. Now I want to talk about some of the personalities involved. For the first couple of centuries, the people who were describing plants were Europeans or North Americans, but we're being replaced largely with Latin Americans. All of the people in this picture, except for Justin Stolten, who's a graduate student at Cornell University, are associated with the Missouri Botanical Gardens. And Mike Graham's in his 70s, I'm 86, and Keating is already retired, so we'll soon disappear from the scene, and we'll be replaced by people like Monica Carlson, who's working on the phylogeny of Anthurium. Mexico and Middle America and Central America are reasonably well represented. 2 people in Mexico, 1 person in Panama, Ortiz, and 1 person in Costa Rica, Marcos Cedeno. And Jay Vanini is a friend of mine who works extensively in Guatemala. The Brazilian aroid researchers are certainly the largest group in North America, or in the Americas. Many of these people were younger students when I first got into botany, but now they're mostly professors and have their own students. So the future of aroid taxonomy in Brazil is very high. They mostly work with, except for Eduardo Gonzalez, they mostly work with Anthurium section Urospadix, with Philodendron, with Heteropsis, and with section Peccaneurium. These are some images of some of the Colombian individuals who are aroid researchers. The Colombians represent the next largest group of aroid researchers. They work over a wide spectrum of plants genera. Marcela Mora is associated with the Missouri Botanical Gardens, but she's done all of her work in Colombia, and she's a specialist on Philodendron. There's also a number of very important students who are going to add to the ranks of specialists in Colombia, and these 2 fellows here and the third guy in the middle are all very important graduate students. Now, I'm just going to list the people who are still active doing research with neotropical plants, and these are Europeans, Americans, Brazilians, Colombians, everybody. So that's not a very long list. And yet we have good potential for doing a good deal of work. Now I want to deal with the taxa. The Neotropical Araceae, 44 genera. I realize you can't read all these names, but the ones in red are all genera with less than 8 species, and they're pretty well known, so I'm not going to discuss them any further. The group that is left over here, 15 genera, are the large to medium-sized genera. And if you'll notice that the two highlighted in red, Anthurium and Philodendron, if the potential number of species is reached, this will constitute almost half of the total number of species of Araceae in the world. The first row of figures are the known number of species. The second row of figures are those that I predict will be found. These are just some of the genera that have been revised and who revised them and when they were revised. Except for Adelonema, most of these revisions, and Caladium, most of these revisions have already been published. All of Central America is finished. So when I talk about something needing to be done, it's almost always South America that needs to be worked on, because we have a checklist for Central America. All of the manuscript for the entire family are in press. It's going to be published in a book in Spanish called The Flora of Mesoamerica. And then for the most part, I'm publishing these as separate English language editions, also with a lot more images, because Flora Mesoamerica doesn't have any images at all. The principal genera that are left to be done in South America are Anthurium, Dieffenbachia, Monstera, Philodendron, and all the remaining in this list. The genus Xanthosoma, actually, we've published all of the species in four revisions, but we ignored Brazil, because at the time Eduardo Gonzalez was working on Xanthosoma in Brazil, and I didn't want to interrupt his studies. Now, getting back to Anthurium, it's the world's largest genus in my opinion, and we have 1,346 species that are already published, but I think the potential is 3,000 or more. In fact, when I add up all the names of species that I have named and described, I haven't necessarily yet tested them in Lucid, but it totaled like 3,500. So I think it'll very easily become the largest genus of plants in the world. The number of people I've worked on is pretty extensive. My Edison did a revision of these palmately lobe things about 25 years ago. I've revised quite a number of different groups, and Monica Carlson and I worked on Spathiphyllum. And in Brazil, both Marcus, Livia Temponi, and Mel Camara all work with Anthurium, either section Pecanerium or section Urospadix. The largest remaining problems is Anthurium. Of course, because of its size, it overwhelms everything else. So the number of unknown species of Anthurium in South America probably exceed all the rest of the species that I'm mentioning put together, because we have only 4 of 20 sections revised, and every time you revise one of these sections, it doubles, triples, or quadruples in size, so that even if I estimate it as 300, it's going to be 500 before I get done. The genus that is really, to me, very exciting is Anthurium section Porphyrochaetonium. I'll show you that just a little bit later. These are sections that are being worked on now, being revised at this time. And the ones that are in red are sections that have already been revised and published, and the ones that are in black are those sections that we are currently working on. And since these are ranked by size, you can see that they start off very small. Most of the genera on this page are relatively small genera and will be easy to revise. But notice the size of these things as you climb up the ladder here. Many of these genera have more than 200, 300, 400 species. And we're just now finishing a Cardiolontium revision, which is that last one in the list, with 377. And that's before we've written the key. So we probably will end up with a lot more species in the end. But it will total perhaps no more than 400 species. The genus, the section that I really wanted to bring to your attention was Porphyrochaetonium. When Engler worked on this, it was one species, and I suspect it has 1,000 species. So that's the sort of climb in size that you see with Anthurium. Now, Philodendron is a group with 482 species published. There are nearly 700 that have been described, and some of these are not yet in the Lucid Key, which we have to do before we can test them. But we'll end up with 1,500 species at least. George Bunting, working in Venezuela and in Central America, did a lot of the early work on Philodendron. Simon Mayo published the subgenus Mechanostigma. I published the subgenus Philodendron for Central America. and Graham did Terramicium. Marcella Moore was important because she wrote the first Lucid Key when she was working at Kew on a Clarf grant. The Brazilians, Cassia Sagorge, is working with Philodendron section Macrobellium. And I wanted to mention especially these last two individuals who are currently working on Philodendron phylogenies at Berlin. Marco Cedeno sitting here in the audience who will give a talk, and Orlando Ortiz. The next biggest obstacle would be Philodendron. Over the past few years, we've described several hundred of these species, but it's just a drop of the bucket. I suspect that this will go on for a very long time. Engler's classification has been, I use it because there's nothing else, and it was based on mostly on the structure of the ovules and to some extent on blade shape. But you can't easily predict a plant just by looking at it, what section it is. You've got to dissect the ovule, so it's not a very good character. With Anthurium, any section I can look at and say what it is, but with Philodendron, you got to dissect the ovules. That's pretty nasty way of determining plants. But we're hoping that these modern phylogenies will come to some better conclusion so that they provide sections which can actually be recognized with the human eye. I want to mention a few other genera. Spathiphyllum was a medium-sized genus with 103 species. I think it's easily revisable within the next year or so. Monstera was revised in 1977 by Madison. Madison. And right now, it's being worked on by Orlando Ortiz and by Alejandro Zuluaga, who's helping with that revision. It's South America that's left. These are some of the genera that have been revised in recent years. I say recent, 2004. My student Guanghua, who is now already passed away, unfortunately, did this revision with Dracontium and I. And the revision of Chlorospatha was published in 2015. It's a relatively small genus, 68 species, but a strange one because every species is known from a different place and you never see it anywhere again. So the potential for numbers of Chlorospathas probably is endless because they all seem to be just in one spot only. And then the genera that need a lot of attention, medium-sized genera, include Dieffenbachia, which is not known well for South America. I've written a key to the South American species, but only based on herbarium specimens at the Missouri Botanical Garden, so it needs a lot more work. And we've described a lot of Stenospermations, but they've not yet been tested in the Lucid Keys, and we're having some difficulty finding the old type specimens to be able to enter some of those species that are very poorly known. A couple of small genera, including Caladium and Urospadix. Alejandro Zuluaga is working on Caladium right now, and there's a student named Oscar Lopez in Florencia who's going to be working on Urospadix. I'm hoping that they can knock these two small genera out. They're always complicated because every genus is endlessly complicated, and you have a hard time finding the original material. Now, I've mentioned these Lucid Keys several times, so I want to explain what this means. Lucid is a company in Australia. It was originally started by a university team, and it is a program, a matrix key, where you use about 200 characters, and you describe all the details of every species into this Lucid Key. And then you do that with every species. So once you get the key fabricated, all you do is choose a number of conservative characters. Usually 6 or 7 characters will take you down to 10 species. And then those 10 species, you want to be sure that you look at them and make sure that they aren't what you're dealing with. Usually I know most of the species that are in the list, but then I have to look up the others. So I've got a determination tool where in a couple of seconds I can pick up the type or the picture or description of any species of plant. So it's very quick to sample these species that are predicted by the key. As I mentioned, you have two views when you start this key. One is the entities, which is on the right-hand side. That's the species, and the other side are the characteristics. And if you choose the conservative characteristics, you can invariably come up with an identification or find out that it's new. If there's any doubt, you can try any way you want to. It still comes up with the same answer. It doesn't exist. So you can try 15 different ways, and it'll still be the same resolution. All right, I got one more slide that I just overpassed, I think. Anyway, I don't need to talk about it because it's a subject of other talks here today, and that is pollination biology. We have a fairly large group of people who are studying the pollination of plants. And that group is really discovering a lot of very interesting things. It isn't that we're finding out the pollinators of a lot of plants, but rather that they're finding very interesting features about these plants, different kinds of pollinators and so forth. So this group is a long way from knowing what pollinates even a fraction of the aroids, but it's discovering a lot about the habits of insects and other organisms that manage to pollinate plants. With that, I leave you with my last picture is, what happened to my picture? Anyway, it was a picture of a bunch of plants that I described. No, it doesn't seem to do anything. Maybe I kicked something out here, but anyway, it was just a picture of a bunch of pretty plants. And they were all species. I've described over 3,000 species, and this was just a handful of them that I had described, just to show you that aroids are pretty and that they're very exciting. My first work with Araceae was on Barrow, Colorado Island, where I come to love the species that grew there. And they were all very boring things, but they were very complicated. And I had to spend so much time working with them that I grew fond of the family. There were no specialists, and everything looked like a species. But with aroids, there's about 15 different views from a species because they have all these heterotrophic forms. And it was because of this that I grew to work on Araceae for the rest of my life. So I'm going to stop there.
You still have 2 minutes. If you want
I'll answer some questions and I can.
Okay.
All right.
Thank you.
If anybody's got a question.
We have some time for a couple of questions. Anybody?
Tom, are you gonna make available those keys at some point?
Yeah, all of the keys are going to. I can't publicize them now because I've got about 1,000 species in there, and if I put them on the web, it's just a matter of going through the characters and writing a description and publishing it. So, no, but in a couple of years I'll be gone, and those keys are all going to be posted on either the website of the Missouri Botanical Gardens, if they will do it, or certainly the International Aroid Society, who wants to do it. So they'll be available. But one thing you got to remember is, just because a key is available doesn't mean that it's always going to work, because we'll still have species that need to be added to the key. So if you come to rely on a key that doesn't have all the species in it, it's not going to work. And I don't think the International Aroid Society realizes that just because they got the key, that it will forever work. You have to have somebody devoted to going in there and adding the other species. And so far as I know, they don't have such an individual. But nevertheless, they will be made available, every one of them. We've got keys for most of the neotropical things. And in fact, I'm going to help with Marco, and maybe hopefully you as well, to build this Monstera key. It'll be easy to do using Anthurium as a base because they have similar features. So yes, it will be.
Mm-hmm. Is it similar to Delta from Pancreas?
Yes, it is.
Which one is better?
Well, I've never used Delta, and I don't know anybody who is using it, so I can't really predict which is better. I think they both work in a similar fashion. I didn't use Delta because somebody told me that it was problematic, and the fellow who was trying to get me to use Lucid was more powerful persuader. But you'd have to pay for it. That's the unfortunate thing. Delta, I think, was free. But also it seemed to have less support. That is to say, if you needed some help in operating it, you had to find somebody else to operate it because the people who created it weren't going to help you. That's what I was told. Any other questions? Yes.
So this is just a question that I have to ask you. You were talking about this last slide, and you made a comment about how you decided to work on this because of the variation you saw, and it is just very attractive to you, and it asks you, you thought about so many questions. And so I just want to know, what is the most exciting thing that you found? And do you have any regrets having chosen such an incredibly variable group of taxa that has
Well,
taken your entire career to get your
Well, I
monogram?
only regret that that I'm not going to live longer, because there's a hell of a lot more to be done, and it's going to require, somebody. It's not going to be done by a committee, unfortunately. My institution thinks that we need to form committees and do work, but committees don't get things done. It's people grinding away at the desk, at the microscope. That's how things are done, and it's going to require, a number of individuals to get to know the plants, because it takes a long time to get to know them. I can go through a forest anywhere, and I can recognize new species left and right, and other people can go there and they just see green things. They don't even realize that they're aroids. Even aroiders sometimes don't see the things. I went to Hatun Satya and I walked on 2 trails, about 400 meters total over the course of 2 days, and I added 20% more species to the floor of Hatun Satya, and they'd been working on that for 6 or 7 years, because they weren't recognizing sterile plants. Whereas when I was walking along, I'd see it as a species I didn't know. I knew it was a Syngonium, but I didn't know which species it was. I assumed it was new. And that's very important. It's important really to have specialists go to the field. We're in dire need of more exploration. They think exploration is over with. Our institution no longer even supports collecting. They don't want us to go to the field. When Peter Raven was in charge, if you weren't in the field, he wondered why weren't you in the field? But now they don't want us to go to the field. And as a result, there's going to be thousands of species go extinct before we ever lay eyes on them. I estimate that 5,000 undescribed species in Colombia alone, just in Colombia, that's more than they exist in the whole world right now. But it's got to be the case. Either that or they're extinct. But even if it is, I'm comparing it with Panama. Panama has almost the same number of species as Colombia, and yet Colombia is 15 times bigger and 10 times more diverse. Anybody who goes from Panama directly into Colombia could see the startling difference in the number of species per unit area. So that was my main regret, is I can't finish it. But hopefully we got people like Alejandro and others that will get an opportunity to go to the field. You've got to go to the field. I don't want to be replaced by a molecular biologist. I mean, I love them, but we need an explorer. We need a person who's willing to go out there and get dirty and climb trees and do the work. Any other questions?
There's one more.
Hi, Tom. Hello. Thanks. With regards to your last point about the need for exploration and field work, do you have any thoughts or maybe even advice as to how to make the argument either to governments or other kinds of institutions, universities, whatever else, to support this kind of work?
Well, I mean, I give you advice, but I don't think any of these arguments will help. Every time I go to a country, I say, you're letting all this stuff disappear, burn up, because it's illegal to collect the plants. But they'll burn it, they'll plow it under, or mine it away, but they won't give me a permit to collect the plant. So it's kind of silly, but it's not my rules. And I think that, unfortunately, nearly all Latin American botanists work for the government. And if the government says, you can't do this, well, they don't do it, because they can't afford to lose their jobs. So they can't afford to rock the boat much, and therefore it's not going to easily change. Conservation will only go so far as saving plants, because most of the land is not in the hands of parks and stuff. If you look at the square footage of South America and the parts that are green, we've only looked at about 1/100% of the total space. And yet with aroids, you could go to a space this size and find 4 new species, and go a kilometer down the road and find another 4 new species. And you can do that for kilometer after kilometer after kilometer. So every little bit of forest could potentially have new species in it. So we really haven't even begun the process of discovering plants. And yet, as far as they're concerned, it's over. Sit here and make these lists and fill out this report and do this and do that and all the kinds of stuff that will end up with no net result when we should be, doing real science. Any other questions? Yes.
All right, so this is our last question.
As we met last year in Aquagenera,
Mhm.
you see that now they changed from orchids to aroids and these kinds of things.
That's some of
Which
my
are in-
influences.
Do you think they can play a part in discovering new species? And
Absolutely, they do. Nearly all the stuff that they're selling is new. It's startling. But this is also the problem. See, it should be science-based. That is to say, they should be giving specific locality data. Maybe they don't need to publish this, but it should be recorded, because when you're dealing with something of unknown origin, you have no idea where it came from, you're reluctant to describe it. Of course, if it's remarkably distinct, you can probably say, well, anybody recognize this if they stumbled on it. But it's, yes, Ecuador and see, Colombia doesn't have such an organization. And they're losing out. They're losing out millions of dollars in actual lost costs, plus the plants are disappearing too. And when they sell those plants, they replicate it about 1,000-fold. So the plant goes all over the world. Everybody's got it. Somebody's going to grow it successfully and spread it to other people. That plant might become the most common plant in the world, whereas it's extinct in the wild. No, I think absolutely they're losing the boat on this subject of trying to save plants. And if I bring it back, I get fired. But I say, well, this was in a fire. They were bulldozing the road. No, pardon, just leave it there. Let it die. And they will go extinct. There's at least 5,000, as many more even possible. And they're not going to be collected, because who's going to collect them? They won't let anybody collect. Very few people collect. And we need a lot more collecting done, because we're just scratching the surface, sort of going to the same spots again and still finding new species. But it's not as if we really made an effort to go out and comb through the remaining forest and find the new species. That's why it's important to have experts do the collecting. And most experts are old and sitting behind their computer doing stuff, because they can find the new species so much more efficiently than a person who doesn't know anything about the group. And this study in England that showed that of all the species described, 3% of them, they were collected and named by 3% of the people, or at least named by them. 3% of all the collectors named almost all the new species. And they had the same characteristics. They worked a long time on a group. They had a group that they was driven to go to the field. Some always took them back there because they were never done. And they visited many places. So those three characteristics, everybody in that group, I call them mega collectors, but everybody in that group was very successful at finding new species, because when they were in the forest, they just fell onto plants, There are people who see plants, and there are people who can walk all day and see nothing. But it's important that older, experienced people continue to work as collectors if they can. At least get somebody with a chair and carry them through the forest. But they ought to be out there with their eyes on the vegetation.
Tom, I'm sorry, we're out of time. Thank you.
Thank you. You're welcome.
Thank you.